Hi, my name is Abbas and I'm going to talk about the identification, classification, and prioritization of the most influential players in normal biological processes and diseases. The ECCB or Experimental Data-Based Integrative Ranking is a classification and prioritization model that entails several novelties. These novelties include independence of any prior knowledge, external sources of information, or training set. So we can say that ECCB is able to prioritize features from, for example, a biological process or disease that there is no prior knowledge regarding that. Also, ECCB is able to discriminate between drivers, biomarkers, and mediators. The ECCB model is composed of two major sections, as you can see in this photo. These two major sections include multi-level filtration and scoring, and classification and integrative ranking. In the first section, supervised and unsupervised machine learning models and network analysis techniques are recruited to generate 13 different scores. Then, in the second section, different combinations of these scores are integrated to classify the features and prioritize them within each class. We evaluated the performance of ECCB model in the identification and prioritization of drivers, biomarkers, and mediators in comparison with contemporary methods. The pipeline on the left-hand side of the screen demonstrates the methodology applied for defining the true positive and negative sets for driver testing. According to the receiver operating characteristic analysis, ECCB outperformed all the other benchmark methods in the prioritization of true drivers across two single-cell RNA-seq, 4 bulk RNA-seq, and a microarray dataset. Similarly, the pipeline on the left-hand side of this slide demonstrates the methodology applied for defining the true positive and negative sets for biomarker testing. The analysis indicated the superiority of ECCB in comparison with all the other benchmark methods in identification of correct biomarkers across 3 disease datasets. We also applied the ECCB model on the entire lung adenocarcinoma dataset to see if ECCB is able to identify both known and novel biomarkers. Also, the results were confirmed at the protein level using immunohistochemistry data obtained from the Human Protein Atlas database. As you can see in this photo, the top 5 biomarkers that were common with the biomarkers previously obtained from the National Cancer Institute database of biomarkers had high ECCB ranks, and their differential expression was confirmed based on the immunohistochemistry data. Also, amongst the top 5 predicted biomarkers by ECCB, the differential expression of 4 of them was confirmed at the protein level. This suggests that ECCB is able to identify known and also predict novel biomarkers of biological processes and diseases. Then we evaluated the performance of ECCB in identifying the mediators. Firstly, we compared the performance of ECCB in comparison with the Melanie model. Melanie is a machine learning-based model for the identification of the mediators of cancer progression. According to the analysis of a breast cancer dataset, ECCB clearly outpaced Melanie in the identification of mediators of breast cancer-associated biological processes and KIK pathways. Then we applied the same methodology on the other disease datasets, and this showed that a considerable proportion of ECCB-derived mediators are involved in the biological processes and pathways of studied diseases. We also developed a function for the visualization of ECCB results. This figure illustrates the top 5 ECCB model-based prioritized genes of the Cancer Genome Atlas breast cancer dataset. To summarize, we can say that ECCB is a one-stop and versatile model for the classification and prioritization of biological entities. Also, ECCB can be applied on any type of experimental data that has at least 2 biological conditions or cell states. Also, ECCB outperformed all the other benchmark methods in the identification and prioritization of correct biomarkers, drivers, and mediators of biological processes and diseases. On top of that, ECCB proposed a new class of genes termed mediators, which would enhance our understanding of the development of normal and disease processes. The ECCB algorithm as well as its visualization function are available in the R package Influential, which is accessible on its GitHub repo as well as the Ground repository. Also, the R package Influential has a comprehensive vignette including video tutorials regarding how to run the XSEER model and how to visualize the results. Additionally, XSEER and its visualization function are available as a user-friendly Shiny app on the Influential software package web portal. At last, I would like to highly thank my supervisors and those who helped me in this project.